ሄሎ እንዴት ናችሁ? ኤቭሪባዲ እንግዲህ ጀነቲክስ 11ኛ ክፍል ጀነቲክስ ምን ላይ ነን? ሜንዴሊያን ጀነቲክስ ባለፈው አቅርቤላችኋለሁ ቪዲዮ። ከሜንዴሊያን ጀነቲክስ ቀጥሎ ኒዮ ሜንዴሊያን ጀነቲክስ ዛሬ የማቀርብላችሁ I am going to give you some know-how of what is there in non-Mendelian genetics or inheritance. And let's go straight into the lesson. NeoMendelian genetics, also known as post-Mendelian genetics or inheritance, expands upon Mendel's original principles by incorporating concepts such as gene interactions, multiple allelism, and polygenic inheritance. Such concepts are concepts studied after the work of Gregor Mendel. Therefore, this is the work of NeoMendelian geneticists, those personalities who came and built on the work of Gregor Mendel, of course. And while Mendel's work laid the foundation for understanding inheritance through dominant and recessive alleles, NeoMendelian genetics addresses more complex patterns of inheritance that involve multiple genes and their interactions. You see, when we talk about Gregor Mendel, let me remind you just when we talk about historical history of genetics, we divide the history into pre-Darwinian, pre-Mendelian, Mendelian, and post-Mendelian. Pre-Mendelian genetics is a dark age of genetics. There are many speculations, so-called theories, hypotheses, whatever. No experimental evidence. And for the second, the cornerstone is Gregor Mendel's work on Pisum Sativum. And then those who came after Gregor Mendel built based on the works of Gregor Mendel. They built the present day modern genetics also. Therefore, NeoMendelian genetics addresses more complex pattern of inheritance that involves multiple genes and their interactions. Even now, genetics is growing at molecular level. We can talk about DNA, we can talk about cutting a piece of DNA or genes and transferring it into another organism. Even we can edit genes to correct mistakes or to enhance phenotype of an organism, or many other things can be done by changing the genetics by gene editing. Key concept, what are the key concepts in NeoMendelian genetics? Key concepts ምንድን ናቸው? key concepts አንደኛው ስለ incomplete dominance, Gregor Mendel የነገረን just one type of dominance, that is complete dominance. Now we talk about incomplete dominance in NeoMendelian genetics. The second, we talk about codominance. What does mean codominance? The third, we talk about multiple allelism. And the fourth, we talk about epistasis or gene interaction, when different genes, not alleles of a gene, different genes located on the same chromosome or different chromosome interact in such a way that they either influence each other in a positive way, or one influencing the other, enhancing the activity of the other or decreasing the activity of the other. Therefore, this is epistasis. We talk also polygenic inheritance, characters that are under the control of many genes on different chromosomes, of course. And these are points which were not discussed or seen by Gregor Mendel. Some of points in NeoMendelian genetics. Now, first let's see incomplete dominance, also known as partial dominance or blending inheritance. I've told you the two different alleles of one trait interact in such a way that the heterozygous individual have an appearance somewhat in between the phenotypes of the two parental varieties. You see, both alleles, the two different alleles of one character, express partially, and they mix up their gene products, and the organism will have a blended phenotype, a mixed phenotype of the two traits. A particular example is flower color inheritance in the 4 o'clock plant. This plant is given 4 o'clock because of the fact that they usually bloom around this area, around this time in the day. And flower color, red and white. When you mix red and white, you will have red allele R, W allele, and the F1 generation are all pink. You see, all these are pink. Produces the F1 generation, all RW pink. If you multiply, if you cross the F1 with each other, it gives you this pink with pink. It gives you one red, two pink, and one W. And produce F2 generation in 1:1 both as phenotypic and genotypic ratio. You see, therefore, this is one typical example of blending inheritance or partial dominance or incomplete dominance. Try these questions. It says, try these questions. In a plant species, if the B allele, blue flowers, and the small b allele, white flower, are incomplete dominant, that means capital B, small b is light blue, the blended light blue. When white and blue are mixed, light blue. What offspring ratio is expected in a cross between blue flowered with light blue flowered plant? 3. What would be the phenotypic ratio of the flowers produced by a cross between two light blue flowers? This is similar with the former one, the previous one, the freezing that 4 o'clock plants. Work for them yourselves, please. It's very simple. Very simple. Okay. In question 2, what offspring ratio is expected in a cross between blue flowers? Blue make blue is capital B, capital B. Light blue is capital B, small b. Mix this. Okay, the second example of NeoMendelian inheritance is codominance. Codominance is genetic phenomenon where two alleles of the same gene are expressed equally in an organism, resulting in traits associated with both alleles being displayed simultaneously. Both alleles expressed fully, so that the organism will have both traits. Since it doesn't follow Mendel's inheritance law, it is called a NeoMendelian inheritance. Yes. The human ABO blood group is one case of codominance. We can see codominance between these two alleles, IA allele, the A allele, and the B allele. The ABO blood group system classifies human blood into 4 main types, 4 main phenotypes, A, B, AB, and O, based on the presence or absence of specific antigens or red blood cells. Of course, this classification of blood type is because based on antigens, A antigen and B antigen. Antigens are protein or glycoprotein molecules inserted and found in the membrane, cell membrane of red blood cells. If the blood contains A type of antigen, that blood is A type. If the red blood cells contain B type of antigen, that blood is B type. If the red blood cell membrane contains both types of antigens, it is AB. If it lacks both types of antigens, it's O. 2 antigens AB, that is. And these antigens are represented by 3 alleles, the IA, the AB, and the IO, or simply A, B, and O, you can call them. And in human, there are 4 blood types, A, B, AB, and O. Blood type is controlled by 3 alleles, as I've told you. This is one case of multiple allelism, we shall discuss later. IO is recessive. 2 O alleles must be present for the person to have type O blood. IA and IB are codominant. If a person receives an A allele from one parent and B allele from the other parent, the blood type will be AB type. This is the case. Type A could be IA or IA-IO. Type B could be IB or IB-IO. O is missing in this. Of course, it is possible to write also in this form, small I. Type O is IO or simply small I, or simply OO. You can make it. Type A has antigen A on the red blood cell membrane and and also when the red blood cell membrane contains antigen A, the plasma or the blood serum contains antibody B. That means A blood type contains antibody B that can destroy B blood type if it comes in. because B blood is not part of the A type of blood. Type B blood has B antigen and A antibodies, anti-A antibodies, A. So as antibodies try to destroy those red blood cells which contain A antigens. Type AB has both A and B antigens, so no anti-A, no anti-B, because both antigens are self. Type O has no A or no B. It contains neither A nor B, so it contains both anti-A and anti-B antibodies, you see. Therefore, if you give A type of blood for O, it will not accept because it contains anti-A. If you give B type, it contains anti-B, it destroys. Therefore, if you give AB contains both types of antigens, antigen A, antigen B. Therefore, even two enemies are there for O blood. Therefore, the reaction, the agglutination reaction is very severe in this case. So, blood type O only accepts from blood type O, but it can give to any type of blood. This is diagrammatic representation. You can see A blood type in the cell membrane contains what? Antigen A. Group B contains antigen B. Group AB contains both antigen A and antigen B. Group B contains no antigens in the cell membrane. But in terms of antibodies, in the serum or the plasma, A contains anti-B antibodies. B contains anti-A antibodies. AB contains no antibodies because both are its own. How can it counter or negate its own blood? O contains both antibodies, antibody A and antibody B. Antibody against B and antibody against A. Antigens in red blood cells, these are antigen A, antigen B, I have told you already. O blood type contains no